Usually when we picture DNA, I think we all see the exact same thing in our heads, right?
Oh, yeah, the classic double helix.
Exactly.
Yeah.
It's clean, it's elegant, it basically looks like a perfectly twisted ladder. But imagine taking the DNA from just one of your microscopic cells and like stretching it out end to end.
It would be massive, well, at that scale.
Right. It would be about 2 meters long. So the real mystery isn't just what the ladder looks like. The mystery is how you cram 2 meters of that ladder into a cellular nucleus that is a fraction of the width of a human hair. And you have to do it without it becoming just a completely tangled, useless mess.
Yeah, because if it becomes a tangled mess, well, the cell dies. It's really not just a packing problem. I mean, it is an access problem.
Access.
Yeah, think of it like this. The sequence of the DNA is the alphabet, but the physical 3D fold, the actual shape, is the grammar that gives it meaning. The physical structure literally determines which genes are turned on or off at any given moment.
Which brings us to the mission for today's deep dive. We are looking at a massive stack of research today, primarily a huge 2026 study published in the journal Cell by Yizhuo Che and colleagues. And they didn't just look at human DNA.
No, not at all. They mapped the 3D genome architecture across 1,025 different species.
1,025. That's insane. So we are tracing 3.8 billion years of evolution today to see how plants and animals took completely different paths in folding their DNA. And we're going to see how you listening to this right now, literally mirrored this ancient evolutionary split inside the womb before you were even born.
It's wild. The overarching finding here, it completely reshapes how we think about biology. What's fascinating here is that early life forms basically had completely random, disorganized DNA shapes. But as complex life evolved, it figured out two, and honestly only two, master blueprints for folding that 2-meter string. Okay,
but before we jump into those two blueprints, I need to understand how anyone actually sees the 3D shape of a genome across 1,000 drastically different species. Like, how do you even build a map of something that small and complex?
Right. So the core technology making this possible, it's called Hi-C, or if you want the full name, chromatin conformation capture.
Hi-C is definitely easier to say.
Much easier, yeah. So picture that 2-meter string of DNA wadded up inside the nucleus. Because it's crammed in there, different parts of the string are constantly bumping into each other.
Sure.
So Hi-C is essentially a chemical trick. Researchers use a chemical called formaldehyde to freeze the DNA right where it sits.
Wait, formaldehyde? Like embalming fluid?
Basically, yeah. It acts like a glue. It instantly cross-links any pieces of DNA that are physically touching in 3D space, even if those pieces are, say, millions of letters apart on the actual unwound string.
Okay, so you freeze it, you glue it together, but you still can't see it with a microscope, right? Okay. So how do you map the glued parts?
Well, they take the whole thing and cut the entire frozen genome into tiny, tiny pieces. Then they sequence those glued together chunks. And every time they find 2 pieces glued together, they mark a point on a massive grid.
Oh, I see.
Yeah, and if you do this millions of times, you build a contact map. It's basically a heat map showing exactly which parts of the genome physically interact most often.
Okay, but I'm looking at the scope of the sources here, and the sheer scale of doing this across 1,025 species, it just seems impossible. I mean, we're talking about genomes as tiny as 2.1 megabases, like this microbe here, Thermococcus codicrentis, which apparently lives in boiling water.
Yeah, tough little guy.
Right. And then they compare that boiling dot to common wheat, Triticum, which is 14.6 gigabases. Wait, that means wheat has a genome roughly 5 times larger than a human's.
It does. Plants are incredibly complex genetically.
So how do you compare a contact map of a tiny microscopic dot to a massive plant like wheat?
That extreme variation in size is exactly what exposes a fundamental flaw in standard Hi-C maps. They are completely blinded by short-range contacts.
Blinded how?
Well, because DNA is a continuous string, right? So any given piece of DNA is obviously going to be touching the piece immediately next to it on the chain.
Oh, obviously.
Right. And in a massive genome like wheat, these immediate neighbor interactions generate so much background noise on the contact map that it just drowns out the vital long-range interactions. The pieces that are far apart on the string but loop all the way around to touch in 3D space.
Okay, let's unpack this. That makes me think of, like, trying to understand the layout of a massive city while standing on a crowded sidewalk.
Oh, that's a good way to look at it.
Yeah, like you can see your immediate next-door neighbors really well, but you have zero sense of how the whole city is organized.
Yeah.
You can't see the grid system. You can't see the major highways connecting opposite sides of town.
Exactly. And the researchers realized they needed a satellite view. So to get it, they developed a totally new algorithm called NormDis. Right. And its entire job is to mathematically strip away what they call distance-dependent biases.
Meaning the next-door neighbor noise.
Exactly. NormDis utilizes a highly calibrated, scaled sampling strategy. It essentially filters out that overwhelming noise of the immediate neighbors and then reweights the hidden, weaker signals of the long-range chromatin interactions.
Okay, so let's pause and do a quick recap for you listening just to make sure we're all tracking the technology before we dive into the wild biology part.
Sounds good.
So bullet 1, Hi-C is the chemical trick using formaldehyde to glue touching pieces of DNA together to map 3D contacts.
Right.
Bullet 2, genome sizes vary wildly across the 1,025 species, making comparisons super messy. And bullet 3, NormDis is the algorithm they built. It's the satellite view that strips away the short-range noise to reveal the long-range architectural highways.
Perfect summary.
So, for you listening right now, have you ever thought about how zooming out from the microscopic daily details of your own life might reveal massive, hidden patterns you couldn't see when you were stuck up close? Because mathematically, that is exactly what NormDis did for the genome.
It really is. And once that satellite view was switched on, the architectural highways were finally visible. The researchers discovered that across the entire tree of life, complex organisms predominantly rely on one of two completely distinct structural shapes.
And the first architecture is called global folding.
Yes, global folding.
I tried to visualize this when reading the sources, and the best image I could come up with is setting up a massive camping tent.
You know, the physics are remarkably similar, just at the karyotype scale.
Karyotype scale.
Meaning the scale of the entire chromosome. So in global folding, the chromosome doesn't just float around loosely, it is spatially anchored at very specific points.
Like the 10 poles.
Exactly like 10 poles. Typically, it's pinned down at 3 locations. The 2 ends of the chromosome, which we call the telomere.
Telomeres, right.
And the middle section, called the centromere.
Because if you don't pin down the poles of a tent, the whole fabric just collapses into an unreadable pile of nylon.
Exactly. And because the chromosome is anchored at the ends and the center, the Hi-C contact maps reveal these really distinct X shapes.
Oh, wow.
Yeah, the researchers classify these rigid polarized patterns as center end axis or center hole. All the ends of the chromosomes cluster together in one zone and all the centers cluster together in another, and it holds the massive DNA string in a highly stable, overarching global shape.
Okay, so that's the tent. But then we have the second master architecture, which doesn't look like a tent at all. The sources call it the checkerboard.
The checkerboard, yes.
And apparently it's driven by something called chromatin compartmentalization. I definitely need your help unpacking this one because the physics seem totally different from the tent poles.
It is entirely different. The checkerboard architecture discards the rigid tent poles entirely. Instead, it organizes the DNA based on activity. In this model, the genome basically physically separates itself.
Separates itself. How?
Well, all the active, quote-unquote, turned-on genetic regions seek each other out and clump together in 3D space. And conversely, all the inactive, turned-off regions clump together in a completely separate space.
Okay, so it's like shaking up a bottle of oil and water.
Oh, precisely.
They exist in the same bottle or, the same nucleus, but the oil and water separate into distinct modular pockets because similar things attract and different things repel.
And when you look at the contact map of this oil and water separation, the mutual attraction and repulsion creates a literal crisscrossed modular pattern. It looks exactly like a checkerboard.
That is so cool.
Right. Regions from the same compartment interact tightly, while regions from different compartments push each other away. And the scientists actually quantify this. They use a concept called information entropy to generate what they call a checkerboard score, or CBS.
Okay, stop right there. Information entropy sounds like a terrifying term from a graduate physics textbook. What did that actually mean for the DNA?
Haha. It does sound intimidating, but entropy is essentially just a measure of chaos or disorder.
Okay, chaos.
Yeah, so if a genome's interactions are totally random and jumbled up, the entropy is high. But if the active genes are strictly segregated from the inactive genes, the oil and water, the disorder drops.
Got it. So a lower information entropy means a highly ordered, incredibly strong checkerboard pattern.
Exactly. And the researchers also found this physical separation correlates heavily with the actual sequence of the DNA letters, specifically regions with a high concentration of G and C letters, which is known as GC or CpG content.
Wait, so it's the actual sequence of the letters that gives a section of DNA its molecular flavor, I guess, dictating whether it acts like oil or water.
You nailed it. The sequence chemistry creates the affinity. High CpG regions tend to be where the active genes are located, so they naturally aggregate together in 3D space, forming these distinct active compartments.
What blows my mind is that these two architectures, the anchored global folding tent and the oil and water checkerboard, they're mutually exclusive. The structural physics just conflict.
Yes.
You can't really have both at the same time.
No, you can't.
So any complex living thing basically has to choose, right?
Yeah.
Do you want to anchor your whole chromosome at specific structural points to keep it organized? Or do you want to separate your active and inactive DNA into repelling modular clumps?
And the evolutionary tree shows that different kingdoms of life had to make that exact choice.
Right, let's do another quick 5-minute recap for you listening.
Okay. Yeah.
Bullet 1. Global folding anchors whole chromosomes at specific points, like tent poles. Bullet 2. Checkerboard separates active and inactive DNA into repelling compartments like oil and water. And bullet 3. They're mutually exclusive architectures.
Exactly right.
So question for you listening at home or in your car. If you were designing a complex operating system, would you prefer the rigid anchored structure of global folding or the highly sorted modular clumps of the checkerboard? Because nature had to decide.
And here's where it gets really interesting, because early on, there was no choice to make.
Right, the ancient stuff.
Yeah. When researchers looked at the earliest, most ancient branches of the tree of life, the ancestral soup, basically they found a complete lack of high-order structure.
So we're talking about the deep ancestors of all cells here. The single-celled organisms that eventually became bacteria, archaea, and eukaryotes. The sources called them LEC, L-U-C-A.
Right. LUCA is the last universal common ancestor, LEC is the last eukaryotic common ancestor, and so on. By analyzing living proxies for these ancient ancestors, the data shows their genomes were basically random, disordered configurations.
So no tents, no oil and water, just a loose jumble.
Just a jumble. The first major architectural split happened as single-celled organisms evolved. Bacteria largely adopted global folding, and they achieved this using a master protein called SMC, which stands for structural maintenance of chromosomes.
Okay, but how does one single protein organize a whole genome?
Well, SMC acts as a physical motor and a clamp. It literally grabs the DNA string and actively extrudes it into large loops, tying them off at specific points to create that anchored tent-like structure.
Oh, wow. So it's actively pulling the ropes.
Exactly. Now, archaea, on the other hand, developed rudimentary, coarse checkerboard patterns. But the truly profound divergence, the big one, occurred when life made the leap to multicellularity. This is the great split between plants and animals.
Okay, yeah. Let's get into this because plants went all in on global folding, and the sources explain their strategy in a way that is incredibly elegant. It all comes down to how plants prefer to group their genes.
Yes. Plants favor global folding because their unique strategy for regulating genes relies on linear genomic clusters.
Linear clusters, meaning in a row.
Right. If a plant needs a specific set of genes to work together, say, to perform photosynthesis or, to grow a root, it places those genes right next to each other sequentially on the actual linear DNA string.
So if a plant wants to read a specific story, it just reads the book chapter by chapter. The genes it needs are already right there in order.
Exactly. Because the related genes are linearly clustered on the string, the plant doesn't need to fold its DNA into complex, separated 3D compartments just to bring active genes together. The global folding architecture simply keeps the overall chromosome stable while the reading machinery just moves down the line.
That makes total sense.
And this linear strategy gifts plants with something extraordinary. Extreme cellular plasticity. Biologists call it totipotency.
Totipotency. Okay, if you, the listener, have ever cut a single leaf off a succulent plant and stuck it in the dirt, you have seen totipotency in action. That single leaf eventually grows into an entirely new, complete plant.
Right.
The leaf cell suddenly decides, hey, I can become a root cell, a stem cell, a flower cell. It could be anything.
It can do that because its genome isn't locked down into rigid 3D specialized compartments. The global folding architecture keeps the genome flexible. The reading machinery can just move to a different chapter on the string to become a root instead of a leaf.
But animals, meaning you, me, the dog, every invertebrate and vertebrate on Earth, we abandoned the plant strategy. We evolved increasingly strong checkerboard architectures. But I have to ask, why would we give up the ability to regrow our whole body from a single piece? That seems like a massive downgrade.
Well, it's because animals don't group related genes next to each other on the string. We scatter them across the entire genome.
Scatter them. Why?
It allows for more complex regulation. But to get those related, scattered genes to work together, animals must use checkerboard folding to cluster them spatially in 3D space.
Oh, I see. So if plants are like reading a standard book chapter by chapter, animals are like an interactive top-up book. The story only makes sense when the 3D folding mechanics bring a character printed on page 1 physically face-to-face with a character printed on page 50.
That is a fantastic analogy, yes.
Yeah.
And that spatial clustering correlates directly with species complexity.
Really?
Yes, the stronger the checkerboard pattern, the higher the species complexity. And they measured this by the sheer number of different cell types, body length, and the size of the proteome.
No.
This checkerboard architecture allows for incredible functional specialization. You have brain cells, liver cells, and heart cells behaving completely differently from one another because their genomes are physically folded into highly specialized active and inactive compartments.
The trade-off is massive, though. We gain the ability to form complex brains and organs, but we sacrifice the plant's plasticity. I mean, I can't cut off your finger and plant it in the yard to grow a new you.
Thankfully, no.
Right, because your cells are too specialized, literally locked into their checkerboard compartments.
Exactly. That tension between plasticity and specialization is one of the foundational drivers of evolutionary biology.
All right, time to take a step back and view the whole board here with another recap.
Oh, go for it.
Bullet 1. Ancestors of life had unstructured, random DNA. Bullet 2. Plants evolved to use global folding and linear gene clustering, which keeps their cells highly flexible, like the succulent leaf. And Bullet 3. Animals evolved to use the checkerboard architecture and spatial gene clustering, sacrificing flexibility for high complexity.
Spot on.
So, listener, think about your own workflow for a second. Are you a linear organizer like a plant, keeping everything in sequential folders so you can pivot quickly? Or are you a spatial organizer like an animal, bringing disparate things together from all over your desk to complete a highly specialized task?
I love that. And what truly elevates this study from a history lesson into a modern revelation is that this exact 3.8 billion year evolutionary split, it is happening right now in real time inside human biology.
The embryo. This completely broke my brain when I read it.
Oh, it's incredible.
There's that old phrase, ontogeny recapitulates phylogeny. The idea that the development of an embryo replays the evolution of its species. And apparently that happens at the structural level of our DNA.
It does. The researchers analyzed early human embryogenesis and found it perfectly mirrors the evolutionary trajectory. When a human embryo is just formed, at the 2-cell stage, the DNA inside those 2 cells does not possess the mammalian checkerboard pattern.
Which means it looks exactly like plant DNA.
Yes. At the 2-cell stage, the human embryo exhibits strong global folding. And remember what global folding provides.
Extreme plasticity to totipotency.
Exactly. Those first 2 cells need to be able to turn into absolutely anything, the placenta, the heart, the nervous system, the skin.
Right, they can't be specialized yet.
Right. To maintain that ultimate flexibility, they rely on the ancient, highly plastic global folding architecture.
But eventually that embryo has to build a brain and a liver. So how does it transition from a flexible plant-like state to a complex human? Do the tent poles just come down?
That's literally what happens. As the embryo develops from 2 cells to the 8-cell stage and eventually into a 6-week embryo, that initial global folding is actively dismantled. The SMC proteins literally take down the tent poles. Then, a stepwise reconstruction begins. The genome starts separating the oil from the water, organizing into the checkerboard architecture. As the cells divide, cellular lineages emerge. The active and inactive compartments form, locking the cells into their specific, specialized identities.
So we literally transition from a highly plastic, plant-like DNA architecture to a highly specialized, animal-like architecture over the course of a few weeks inside the womb.
Yes.
The microscopic folding process perfectly mirrors the macroevolutionary timeline of life on Earth.
It really does.
Let's just pause. Bullet recap.
Yeah.
Human embryos start with global folding at the 2-cell stage. They transition to checkerboard from the 8-cell to 6-week stage. And this mirrors the evolutionary timeline of life on Earth. Just take a second, you listening, and think about the fact that your own cells had to basically reenact billions of years of structural evolution just to build your body.
It's humbling, isn't it? It fundamentally forces us to realize that DNA isn't just a linear code to be read. It is a dynamic physical architecture that dictates the absolute boundaries of what a living thing can be.
We always hear the scientific adage, structure determines function. We first realized that with the discovery of the double helix. But this study scales that concept up to an unimaginable level. The massive three-dimensional architecture of the entire genome dictates whether an organism is a simple microbe, a highly adaptable plant, or a specialized animal.
And, it raises a profound question about the future.
Oh, like what?
Well, single-celled ancestors started with random, disorganized DNA. Then complex life diverged into the global folding of plants and the checkerboard of animals. But evolution is a continuous process.
Right, it never stops.
Exactly. As human evolution progresses and our cellular complexity theoretically increases over the next million years, is it possible our DNA might discover an entirely third state of high-order architecture?
A post-checkerboard genome, that is wow. What would that even look like under a microscope?
I have no idea.
More importantly, what kind of biological abilities or unimaginable traits would a third architectural state unlock for whatever comes after us? It makes you look at every living thing around you, the trees outside, the dog on the rug, yourself in the mirror, and realize that underneath it all is just a masterclass in microscopic origami.
That's a beautiful way to put it.
Well, thank you for joining us on this deep dive. Keep exploring, keep questioning, and never forget the incredible folding universe inside every single one of your cells.